As part of the AAPS, Educational Resources Committee program, we're pleased to visit the University of Kentucky College of Pharmacy and to converse with Dean Joseph Swintosky, who's widely recognized as one of the leading innovators in pharmaceutical technology and in the development of pharmaceutics. Joe, one of our privileges is to talk to some of the people who were the early pioneers who actually contributed to the advancement of pharmaceutics, especially in the post-World War II era. Primarily that time when product development in industry and when academic research programs, especially at the graduate level, led to the development of pharmaceutics. You were one of those people who spent some time at the University of Wisconsin in the 1940s and 50s, not only as a student and graduate student, but also as a member of the faculty. That must have been a very interesting place to have concentrated so much talent at that time to lead to the early evolution of pharmaceutics. What made Wisconsin so special in the 40s and 50s?
I started pharmacy school there in 1938, and it was pretty obvious to me after some time that the people who were my teachers and leaders in the college were also researchers, and they seemed to be having a real good time doing what they were doing. They were inspirational people, and it's hard to know just when one locks in on graduate education and a career of education and research, but sometime in that period, people like Dr. Uhl and Dr. Parks and Dr. Edward Kramers, some of the people who comprised the faculty at that time, influenced my thinking about graduate education in pharmacy.
What made you seek a graduate degree in the 1940s in pharmacy, in pharmaceutics in particular?
Not only did I have the inspiration of people in the College of Pharmacy, but I happened to be living with a contemporary roommate, Chet Okonsky, who was a chemist and kept talking about going on to graduate school in due time. And it also happened that in 1939, he and I encountered three other individuals living in the same rooming house, Takeru Higuchi, Joe Teresi, and Chet Throckmorton, who had profound effects on our thinking about where we were going in education, in our own personal education. And they talked about graduate school for us.
And what urged you, after you completed your graduate studies, to consider a teaching career at that time?
Well, in entering into graduate study, one of the responsibilities given to me was as a teaching assistant and ultimately as a part-time instructor to recover funds to pay for my education, and it was truly enjoyable. It was an enjoyable kind of life, teaching and doing research.
How did physical chemistry emerge as such an important underpinning discipline in the curriculum that was being developed at Wisconsin in those days?
I suppose I draw on the influences of my chemist friends. It was such a fortunate circumstance for me to be located in a home, in a house, rooming house, that had graduate students like Takeru Higuchi and others to turn to when you had questions about your courses in chemistry. And these people took such an interest in me and my roommate. They indicated that there was much in pharmacy that seemed to be related to chemical engineering and analytical and physical chemistry, but they didn't see any of it in our curriculum. Why didn't we have formal courses in this area? And that was a question that I thought about a lot, as to why we didn't have these courses if these chemists could see that much of pharmacy were pharmacy courses indeed had a basic science need in physical chemistry that was not being taught to us.
It seems that that was a profound influence on you, not only in your thinking, but also in some of your writings. Some of the work that you did on your dissertation on measuring surface areas and looking at particle size behavior of solids turned out to be the basis for a very important chapter that you helped author in the American Pharmacy series. Now, that series of chapters, one of which was written by you, has been identified as one of the early contributors to the evolution of pharmaceutics as a science as taught in various colleges of pharmacy. How did you feel about looking at a physico-chemical problem and then using it as a basis of a chapter? Do you recall the thinking that went on in putting together that collection of papers?
Well, I suppose we have to go back just a little bit. I recall Hiruchi's influence on my reading habits, for example. He would point out books and journals for me to look at, and one of them that I recall very distinctly here was a series of books by Jerome Alexander in colloidal chemistry. I believe that was the title. And there were many chapters in these books written by different authors, and one could become very excited reading through one of these chapters and seeing how principles of physical chemistry were applicable to many natural problems and processes. So somewhere along the line there, that fortified my feelings that I just have to get a background in physical chemistry and more mathematics into the graduate courses that I would take.
You were one of the principal architects who served as a bridge by transferring pharmaceutical science and technology from the academic ranks immediately to industry when you went to SmithKline and French laboratories. How did you find the use of pharmaceutics in process technology, especially in control release, to come about? What were the principal leads that you had in that area?
Well, when I entered into the pharmaceutical industry in 1953 and joined the pharmaceutical chemistry section at SmithKline and French, first of all, I was a little amazed that it was called the pharmaceutical chemistry section, because it was in fact a pharmaceutics research and development section. Its problems were those of creating dosage forms that could be ultimately marketed. And though there were some excellent pharmaceutical technologists in that section, I found that there was nobody there who really had formally gone on to get their PhDs in pharmaceutics and had quantitated what I would say is the applications of physical pharmacy or physical chemistry to dosage form design.
One of your earliest applications in you and the rest of the group that was at SmithKline was toward utilizing some of these principles for specialized coatings, especially to develop control release systems. How did that evolve?
When I arrived at SmithKline French in 1953, they had just introduced to the market a single sustained release product, their Spancel capsule. And so they had the technology pretty well established. They had utilized these small sugar pellets, applied to them a drug, and then coatings of various thickness to effect a sustained release. The science had really not been explored too much, and with their success with this original product, they felt the need to bring in somebody who could study the science underlying this kind of technology, study it, and bring credibility to the idea and to products. So that's kind of where I landed in 1953, and I immediately got assigned to a development project in this area, and within a year or two was spending most of my time working on science rather than on specific product problems, development of products.
At that time at SmithKline, you applied some of the concepts of pharmaceutics to biological systems by especially using the science of kinetics and studying rate processes. How did you evolve the use of the biological half-life to explain the elimination kinetics of a drug? How did those studies begin?
Well, the first product development project I was assigned to was the development of a sustained release tablet form of theophylline or aminophylline. And I would guess that any physical chemist or physical pharmacist assigned to that kind of problem would immediately start thinking about, well, how fast does this have to dissolve and release its drug content before the for this tablet to maintain a constant blood level or nearly constant blood level, and felt that we needed to learn something about the absorption characteristics, the excretion characteristics, in order to do a scientific job in this area. So it was very early on in the project in 1953 that I began looking for some data in the literature on blood levels that are achieved in human beings when theophylline is administered. And having looked at these and seen the fall-away curve for theophylline, decided to try plotting it on semi-log paper because it looked like it might plot as a exponential process, and sure enough it did. So we got onto that very quickly.
Were you able then to use some of the observations you made to design experiments that would allow you to be able to improve and optimize the collection of data?
Oh, sure. We immediately began putting drug theophylline into dogs, and at specific times, and determining the blood levels. We called upon data from the literature to confirm that not only theophylline but a whole number of drugs were apparently excreted by an exponential process, and we would have to figure out ways of designing our dosage units such that the rate of absorption from these sustained action oral dosage forms would just compensate for the metabolism and excretion of the drug to maintain a constant or fairly constant blood level over a 12-hour period. This is what we were trying to do, of course.
By that time, you had crossed over disciplines into the biological and clinical sciences. This must have been somewhat of a new experience to have so many different scientists working together on a team in the project. Was that relatively new kind of thinking in industry at that time?
At that time, it certainly was new for pharmacists to get involved in animal and human studies with drugs. Having come from the academic arena where one isn't constrained in your thinking and what you do, it seems so very natural to move out of the arena of physical chemistry and physical pharmacy and beakers and tablet machines and mixers and that sort of thing, and take these concepts right into animal studies. And at the time, we certainly didn't say we were going to leave what was the traditional physical pharmacy area to go into the biophysical, but it seemed like such a natural thing to do, to start applying these same ideas and principles to studies in animals where it seemed important to get certain data to help in the design of the dosage form.
Well, this early application of pharmacokinetics, if you will, was probably well received within your firm, I assume. But perhaps industry operated more in a proprietary area, perhaps not always sharing this information until it had been exploited and developed into a commercial product. Who did you see as some of the early investigators who were making this kind of science, this kind of application of pharmaceutics, more widely known through their publications or by their presentations at meetings at that time?
Well, in the 1953 to 1956 era, I had the feeling that we were working in this area alone. I wasn't aware of anybody actually getting any laboratory data of the kind we were getting that led to our publications in pharmacokinetics. I started giving lectures in several schools across the country on some of the data that I was observing in the literature. I talked about the illustrations and possible applications of first-order drug elimination from the body, but the company discouraged us from talking about our specific product work until such time as the product was put on the market. So we didn't really publish the data until '56, but we had it for several years before that, and I don't know of any similar kind of studies that were in progress during that time.
The scientists that work with you and you yourself, what kinds of meetings, national meetings, were popular at that time in which scientists from industry and academia came together to share some of their thinking, especially in pharmaceutics and pharmaceutical technology?
There really wasn't all that much that I can recall going on in the way of a close communication between people in academia and the industry. The industry was rather secretive at that time. I think the general expectation was that you would not talk about what you were doing in product development and problems that you encountered in making useful products. That kind of evolved in the over the years as people began publishing. I think if you were to check the literature, you would see almost no publications from the industry on what we might call development or research projects in pharmaceutics. Having come from academia, I felt compelled to continue publication, and I also felt that I might return quickly to academia because I loved it. I came out into the industry to learn something about it, but I had visions of getting back into academia rather quickly. I think when we began publishing and receiving authorization from the company to publish on our products and our product work, this, in fact, encouraged other people in the industry to do the same. So if you check the literature of the 1950s, there wasn't really much published early in the 1950s, but late in the 1950s, things really picked up, and people began having more discourses between themselves from academia and industry.
One of the earlier conferences that brought together people from industry as well as from the academic ranks who were doing research in pharmaceutics were the Land O'Lakes conferences. How did those conferences come into being, and how did you, I know that you were a frequent attendee of those early conferences, how did one find such a cooperation between industry and academia at that time?
I suppose Dr. Louis Bussey and perhaps a few other people, Dale Worster, people of that period recognized that there needed to be more communication between scientists in the industry and academia, and that if there was going to be such communication, it perhaps ought to be initiated by the academic people who had no constraints, and they could see themselves as doing something very worthwhile by bringing about this communication, and it would be good for the educators and it would be good for the people in the industry who are pharmaceutics researchers. So I believe this started someplace in the late 1950s, and it really was something that logically followed the beginning of publication from the pharmaceutics people in the industry in the mid-50s.
Were these meetings very popular with industrial scientists?
I think they were exceedingly popular. It was another opportunity for people from the various companies and from the schools of pharmacy to meet, to talk about the basic science underlying product development. I think that's where the principal emphasis was, and it followed so very logically the introduction of basic science, particularly quantitative analysis and physical chemistry, into the curriculum of the graduate students.
You've spoken much about the basic science component in graduate programming. Did you feel at that time that there might have been too much or not enough emphasis on pharmaceutical technology? There were certainly different opinions as to how practical a graduate program should be to prepare a scientist for using physical chemistry in an industrial setting. What was the kind of thinking that was going on as the graduate curriculum was being developed in those years?
Well, I had reflected previously on the influence of certain chemist friends in my thinking about preparation for pharmacy, and specifically preparation for graduate study. So in 1942, I thought I was the first pharmacy graduate student to start on a program of study including physical chemistry and mathematics. This to be coupled, of course, with advanced organic chemistry and biochemistry. There was much communication going on at that time between myself and Tak Hiruchi, who was not yet in pharmacy, but it was a graduate student. And you have to understand that a person like Tak was somebody I saw every day, several times a day. We lived like brothers. And so he and some of these other chemists were very influential in my own decision to pick up on physical chemistry and math. And my professor, Louis Bussey, obviously was very interested in pharmaceutical technology, and in my discussions with him, it seemed so very logical that we were approaching research problems of a similar type in our discussions. He basically is a technologist, but here I was coming under the influence of other people who were saying, hey, this is a physical-chemical problem.
Dean, what does it take to make a great researcher?
I guess, the people who preceded me at Wisconsin had great influence on me, and people with a certain amount of dissatisfaction with things as they are always looking for better ways of doing things. I was fortunate, I guess, to be associated with these kinds of people who were creative. Their dissatisfactions were, I'm not sure how I would articulate that. They were wonderful people, but people who were always trying to improve upon the status quo. And they were brilliant. They were intellectual kind of people. And so when I had the opportunity to watch Tak Hiruchi do research, and most people didn't have that opportunity as I did. I observed him as a graduate student who could figure out ways of doing things that had not yet been done. And when I would meet with people like Lloyd Parks, or with some of my teachers, early teachers who were working in their on their PhD thesis, Lloyd Bowers and Ed Rowe and Austin Dodge, people like this. They were naturally inquisitive people. They were bright, they were hardworking, and it looked like they were problem solvers. And if you have a great interest in solving problems of your profession, if these are technical problems, perhaps you'll be fortunate enough to find answers to problems that people hadn't found previously, and you have to enjoy this kind of life. I saw my peers and my teachers in this area as making significant contributions to the profession and society because they were naturally creative people. And it's very hard to know why they possessed those kinds of traits in such a mix to make them leaders and discoverers and contributors to the profession in research.
And what inspired you to seek the pharmaceutical career path that you eventually followed?
I, of course, hadn't contemplated coming specifically to Kentucky. I guess I always left the door open to return to academia someday, but in the course of exposure to various kinds of people in my own academic upbringing and my relationship to some of the people, particularly at Wisconsin, I guess I tended to become the research type person, the person who, as a teacher and educator, felt that there were certain things in education that needed improvement far and beyond what was happening in the 1960s when I decided to come back to academia. And when it became apparent that as a dean I could attempt to bring some of this knowledge of biopharmacy and pharmacokinetics into the curriculum, and I could influence the physicians and the vice president here at the University of Kentucky to allow us to use the hospital and patients in our education program in pharmacy, it looked like a real opportunity to do something new again and something worthwhile for the profession. We needed to have a good environment to teach a type of pharmacy that had a clinical emphasis, and that did not exist prior to the time I arrived at Kentucky. And with some assurances that there would be cooperation from the administration, getting these in writing and laying out a plan for the future and getting assurance from the president and the vice president at that time that that was a good way to go to help the College of Pharmacy develop at Kentucky, I decided, well, it's time to go into academia again.
Was there anything especially significant about the Philadelphia area that made it a unique setting for pharmaceutical research and perhaps for dialogue amongst various investigators?
Well, that's an appropriate question, I think, because Philadelphia was, in my opinion, really a leading area in the development of pharmaceutics from an industrial standpoint. At the time that I landed there in 1953, there existed a Philadelphia chapter of the American Pharmaceutical Association, and Dean Sprouse at Temple and Dean Tice at Philadelphia College of Pharmacy and Science had provided room in their respective colleges to meet monthly. And there was an opportunity for people in academia and in industry to come to a dinner and then a program on some aspect of pharmacy. And with the high concentration of industry in that area, it turned out that the industrial people, like myself, and people like Tom Masick and George Schneller, many others who are people who have had impact on the profession, had opportunities to meet each other and began talking about things in formal meetings and in informal meetings at these conferences. We got to know each other quite well, and I suspect that it was one of the kinds of meetings that brought about a better understanding between pharmaceutical scientists and really a pleasurable opportunity to get to know your peers, your counterparts in other companies and in academia. It was a better place to do this than any I'd ever encountered anyplace else. So this group was operational, and when I landed there in 1953, and what they had been doing prior to the time I got there, I'm not sure, but it seemed like a natural place to go every month to know your counterparts in other industries, and I really enjoyed it. And I felt they influenced my thinking and helped in the evolution of what we call today pharmaceutics.
Well, that group was certainly active with the APHA at first, and then the Academy of Pharmaceutical Sciences, and then even now with the American Association of Pharmaceutical Scientists. There was also a meeting that evolved in the 1960s and early 70s, dealing with primarily research. We now know it as the Hiruchi Research Seminar, but its original start really was as primarily a research seminar for industrial and academic scientists. And I recall that one of the meetings, the second meeting I believe, was held in the Philadelphia area at Skytop, Pennsylvania. Didn't you have a major part to do with organizing that meeting, Joe?
Well, the details are fuzzy, but I remember at that meeting in the Poconos that I urged that this meeting become an annual affair and had become known as the Hiruchi Research Seminar. Whether it retained exactly that name, I think it was accepted by acclamation that this should indeed continue, and that conference or seminar has been continued over these many years.
I recall the incident because I was chairing the meeting in which you introduced the motion, and I recognize you from the rear of the room. So that is the specific meeting, and I do believe that is the title of the seminar, the Hiruchi Research Seminar at the present time.